Welcome back to Studious. Today we are going deeper into bone than you have ever gone before. This video is titled The Living Blueprint of Bone, and that title is deliberately chosen because bone is not the static, inert, chalk-white structure that a museum skeleton might suggest. It is one of the most dynamic, metabolically active, and architecturally sophisticated tissues in the entire human body. It is constantly sensing its environment, responding to mechanical forces, repairing microdamage, and reshaping itself every hour of every day, from birth until death. Our title slide reveals the engineering complexity we are about to unpack. Looking at the blueprint diagram, we can already identify multiple structural layers: the cortical bone matrix shown in cyan, the trabecular network in amber, the narrow marrow cavity, the periosteum wrapping the outside, the Haversian canals penetrating the dense cortex, and the Volkmann's canals cutting laterally between them. At the microscopic level, we see the osteocyte lacunae, cavities housing the living cells embedded within the mineralized matrix, and osteoblast activity laying down new tissue at the surface. By the end of this atlas, every single one of those terms will be completely clear. We will follow the architecture from the macroscopic skeleton all the way down to the individual molecules that give bone its extraordinary combination of flexibility and strength. Let's begin.
Before we examine the microscopic details, we need to reset a fundamental misconception that many students carry into their first histology course: the idea that bone is simply scaffolding, a passive structural frame that holds you upright. This could not be further from the truth. Bone is a highly metabolically active smart material, and it performs 5 distinct, life-critical functions simultaneously. First, structural support. Bone forms the adult skeleton, the rigid physical framework upon which all soft tissues are organized and supported. Second, protection. Bone shields vital organs from traumatic injury. The cranial vault encases and protects the brain, the ribcage guards the heart and lungs, and the vertebral column shields the spinal cord. Third, movement. Bone provides rigid anchoring surfaces for skeletal muscle attachments, creating the lever systems that convert muscular contraction into coordinated movement. Fourth, metabolic storage. Bone is the body's primary mineral reservoir, storing approximately 99% of total body calcium and 85% of total phosphate. These minerals are not locked away permanently. They are dynamically mobilized and deposited in response to the body's needs, regulated by parathyroid hormone, calcitonin, and vitamin D. Fifth, hematopoiesis. The medullary cavity and the spaces within spongy bone house the red bone marrow, the tissue responsible for the continuous production of all red blood cells, white blood cells, and platelets throughout life. 5 functions, 1 tissue. This is why bone matters so deeply in clinical medicine.
The first level of bone organization is macroscopic, visible to the naked eye, and the overall shape and architecture of a bone directly determines its mechanical role and its developmental origin. The human skeleton contains 206 bones, and they are classified into 5 categories based on their gross morphology. Long bones are defined by having 3 distinct regions: a central shaft, called the diaphysis, flanked by 2 expanded ends, called epiphyses. They are predominantly found in the limbs: the femur, tibia, fibula, humerus, radius, and ulna. Their length allows them to function as mechanical levers, maximizing the force output of attached muscles. Flat bones are broad, relatively thin plates that enclose defined cavities and provide maximum surface area for protection and muscle attachment. The parietal bones of the cranial vault are the classic example, as are the sternum and the scapula. Short bones are approximately equal in all 3 dimensions: knobbly, roughly round or cubic, and are named for their compact shape. The carpal bones of the wrist, including the lunate, and the tarsal bones of the ankle are the textbook examples. They transmit compressive forces in multiple directions simultaneously. Sesamoid bones are rounded bones that develop entirely within tendons, embedded in the fibrous tissue of muscle tendon units. Their purpose is to increase the mechanical advantage of the tendon, reducing friction and redirecting the line of force.
The patella or kneecap is the largest sesamoid bone in the body. Irregular bones are those that defy the other four categories because of their complex three-dimensional geometry. The vertebrae and many of the facial bones are the clearest examples.
When we cut through a long bone transversely and examine what we find from the outside in, three functionally distinct layers are revealed, each with its own cellular population, vascularity, and biological role. The outermost layer is the periosteum, a double-layered fibrous sheath that wraps around the entire external surface of bone, with the exception of the articular cartilage at joint surfaces. The outer layer of the periosteum is dense fibrous connective tissue, rich in collagen fibers and the blood vessels, nerves, and lymphatic vessels that supply the bone. The inner layer, called the cambium or osteogenic layer, is cellular, containing osteopregenitor cells and osteoblasts that are responsible for appositional bone growth and fracture repair. The periosteum is also the anchor point for muscle tendons and ligaments via specialized collagen bundles called Sharpie's fibers, which penetrate directly into the bone matrix. Clinically, the periosteum is richly innervated, which is why periosteal tearing, as in a fracture or bone tumor, produces some of the most severe pain in medicine. The middle layer is compact bone, the dense, hard outer shell, composed of mature lamellar tissue organized into cylindrical structural units called osteons. This is the load-bearing shell of the bone, constituting about 80% of total skeletal mass. The innermost region is the medullary cavity, the hollow core of the diaphysis, containing yellow bone marrow in adults, a mixture of adipose tissue and blood-forming cells supported by a delicate internal scaffolding of spongy bone.
All bone tissue, regardless of its location or function, passes through two sequential phases of organizational maturity. Understanding this distinction is critical for interpreting both normal histology and pathological states. The first phase is woven bone, also called primary bone or immature bone. In woven bone, the collagen fibers of the matrix are laid down rapidly and without any particular directional order. They run in a chaotic, interlacing pattern in multiple random orientations. This disordered architecture is reflected in the microscopic appearance, irregularly distributed osteocyte lacunae scattered throughout a visually unorganized matrix. Woven bone is mechanically relatively weak. The collagen fibers cannot bear load effectively in any single direction because they run in all directions simultaneously. However, woven bone forms quickly, and that speed is its advantage. It is the emergency response tissue, present in fetal bones during development, and the first bone laid down at a fracture site during early repair. Clinically, the abnormal persistence or excess production of woven bone in an adult is a hallmark of pathological states, including Paget's disease of bone. The second phase is lamellar bone, the mature, permanent form found in all healthy adult bones. Here, the collagen fibers are laid down in a highly organized pattern, arranged in parallel sheets called lamellae, with the fiber direction alternating between adjacent lamellae, much like the alternating grain in plywood. This alternating architecture provides resistance to mechanical stress from multiple directions simultaneously, making lamellar bone dramatically stronger than woven bone. Over time, typically within weeks to months after initial woven bone formation, specialized remodeling cells replace all woven bone with this organized lamellar architecture.
Mature lamellar bone does not exist in just one architectural form. It organizes itself into two very different macroscopic expressions, each perfectly engineered for a specific mechanical role within the skeleton. The first is compact bone, also called cortical bone. This is the dense, highly calcified, solid outer shell of bone, the hard, smooth, white material visible on the outer surface of any dry bone specimen. Compact bone accounts for approximately 80% of total skeletal mass and provides the primary resistance to bending, twisting, and compressive forces. Microscopically, it is composed of densely packed structural units called osteons, or Haversian systems, which we will examine in detail in the next slide. Compact bone has a very low porosity, typically around 5 to 10%, meaning it is almost entirely solid mineralized matrix, with only narrow channels for blood vessels and nerves running through it. The second form is spongy bone, also called cancellous or trabecular bone. Unlike the solid wall of compact bone, spongy bone has a completely open, lattice-like architecture, a three-dimensional network of thin mineralized struts and plates called trabeculae, which are oriented along the principal lines of stress passing through the bone. Between the trabeculae, the spaces are filled with bone marrow. Spongy bone is found predominantly in the epiphyses of long bones, the interior of flat bones, and the core of short and irregular bones. Despite its porous appearance, the trabecular architecture is brilliantly engineered. It provides remarkable strength-to-weight efficiency, allowing the skeleton to be both light enough for movement and strong enough to bear enormous loads.
The extraordinary mechanical properties of bone, simultaneously strong enough to resist shattering and flexible enough to absorb impact without brittleness, arise from its unique extracellular matrix composition. And the key to understanding this lies in a precise two-component system that the slide beautifully illustrates as matrix masonry. The first component is the osteoid, the organic framework of the matrix. Think of it as the rebar inside reinforced concrete. Osteoid consists primarily of type 1 collagen fibers, long, rope-like protein chains with enormous tensile strength. These collagen fibers give bone its flexibility and its ability to resist being pulled apart under tension. In addition to collagen, osteoid contains non-collagenous proteins, including osteocalcin, osteopontin, and fibronectin, which regulate mineralization and cell adhesion. Under a standard hematoxylin and eosin stain in the histology lab, osteoid appears light-colored and somewhat unorganized. You see it most prominently at bone-forming surfaces adjacent to active osteoblasts. The second component is hydroxyapatite, the inorganic mineral component. This is the concrete poured around the rebar. Hydroxyapatite is a crystalline calcium phosphate mineral with the chemical formula Ca10PO46OH2. Calcium and phosphate ions from the bloodstream bind and nucleate onto the collagen scaffold, progressively filling the spaces between fibers with rigid crystal deposits. This mineralization process is what transforms the soft, pliable osteoid into the rock-hard, load-bearing tissue we recognize as bone. Under H&E stain, fully mineralized bone matrix appears a smooth, homogeneous deep pink. The combination of organic collagen flexibility and inorganic hydroxyapatite rigidity is what makes bone the most mechanically versatile structural material known in biology.
The functional and structural unit of compact bone is the osteon, also called the Haversian system, and it is one of the most elegantly engineered microscopic structures in all of human anatomy. Picture a hollow cylinder, approximately 200 to 300 micrometers in diameter, running parallel to the long axis of the bone. At its core is the Haversian Canal, an open central channel approximately 50 micrometers wide, containing 1 or 2 small blood vessels, a nerve fiber, and lymphatic capillaries. This is the lifeline of the osteon, the dedicated blood supply that nourishes the bone cells embedded in the mineralized matrix surrounding it. Because compact bone is so densely calcified that nutrients cannot simply diffuse through it from the outside, every cluster of osteocytes needs to be within approximately 200 micrometers of a Haversian canal, the maximum distance over which oxygen and nutrients can diffuse effectively. Surrounding the Haversian canal are the concentric lamellae, between 4 and 20 concentric rings of mineralized matrix, each approximately 3 to 7 micrometers thick, arranged like the rings of a tree trunk. The collagen fiber orientation rotates between adjacent lamellae, alternating between left-handed and right-handed helical patterns. This alternating arrangement is the source of the osteon's extraordinary resistance to torsional stress. Connecting the Haversian canals of adjacent osteons and connecting the central canals to the periosteum and medullary cavity are Volkmann's canals, running perpendicular or oblique to the long axis of the bone. In cross-section, Haversian canals appear circular, Volkmann's canals appear oval. This is an important histological diagnostic distinction in the practical lab.
Inside every osteon,
embedded within the concentric lamellae, are the most remarkable in all of cellular biology. Osteocytes were once osteoblasts, active bone-building cells working at the surface of the matrix. As each osteoblast secretes osteoid and the matrix mineralizes around it, the cell becomes progressively entombed completely. The small cavity it now permanently occupies within the lamellae is called a lacuna. Each lacuna is precisely positioned between layers of lamellae, visible in a histological section as small, dark, lens-shaped spaces within the pink mineralized matrix. But here is the critical question. How does a cell survive, permanently embedded in solid, mineralized bone with no direct blood supply of its own? The answer lies in the canaliculi, tiny, hair-like tunnels radiating outward from each lacuna in all directions, like spokes on a bicycle wheel. The osteocyte extends long, slender cytoplasmic processes through these tunnels, making direct gap junction connections with the processes of neighboring osteocytes, and ultimately reaching all the way to the Haversian canal surface. This creates a vast intercellular communication and transport network spanning the entire thickness of compact bone. Through these canalicular connections, osteocytes receive nutrients and oxygen from the Haversian canal blood supply, exchange waste products, transmit mechanical signals, and release signaling molecules, including sclerostin, a key.
At the very core of every long bone lies the medullary cavity. And far from being an empty space, it is a biologically rich, highly vascularized ecosystem that sustains life in a way that has nothing to do with structural support. The medullary cavity is framed and supported by the trabecular network, the delicate three-dimensional lattice of spongy bone we examined earlier. These trabeculi do not merely provide mechanical support for the inner cavity. They are dynamically responsive structures, continuously remodeling themselves to align with the mechanical forces passing through the bone. When you exercise and load the skeleton, the trabecular architecture literally reorients itself over weeks to more efficiently distribute those forces, a phenomenon predicted by Wolff's law, first described in the 19th century. Coating the internal surfaces of all trabeculi is a thin, specialized connective tissue layer called the endosteum. The endosteum is the internal equivalent of the periosteum. It contains osteoprogenitor cells, osteoblasts, and osteoclasts, making it the primary site of internal bone remodeling. It is the endosteum that responds first when parathyroid hormone signals the need to release calcium from the bone matrix into the bloodstream. Filling the spaces within the trabecular framework is the bone marrow itself. In adult long bones, the diaphyseal marrow is predominantly yellow marrow, consisting of adipocytes, fat cells, along with a supporting vascular network and scattered developing blood cells. In contrast, the red marrow found in flat bones and the epiphyses of certain long bones is the actively hemopoietic tissue, producing approximately 2 million red blood cells per second in a healthy adult.
The production of bone matrix is not performed by a single cell type. It is orchestrated by a dedicated cellular lineage that progresses through 3 sequential stages of differentiation, each with a distinct morphology, location, and functional role. The lineage begins with osteoprogenitor cells, the mesenchymal stem cells of the bone compartment. These are spindle-shaped, relatively undifferentiated cells that reside along the inner surface of the periosteum, the endosteum, and in the walls of the Haversian canals. They have a very low profile under the microscope, small, flattened cells that are easy to overlook, but they are the essential stem cell reservoir, capable of self-renewal, and when triggered by the appropriate molecular signals, particularly bone morphogenic proteins, BMPs, and the transcription factor RUNX2, they proliferate and commit to the osteoblast lineage. The second stage is the osteoblast, the active bone builder. Osteoblasts are cuboidal to columnar cells clustered on the free surfaces of bone matrix, wherever active bone formation is occurring. They have a large, eccentric nucleus and a basophilic cytoplasm packed with rough endoplasmic reticulum, reflecting their enormous protein synthesis activity. Their primary task is secreting osteoid,
the organic matrix precursor, and expressing alkaline phosphatase to drive mineralization. The third and final stage is the osteocyte, formed when an osteoblast is fully surrounded by its own secreted and mineralized matrix. Once embedded, the cell downregulates its synthetic activity and transitions into a long-term maintenance and mechanosensory role. This is the cell that entombs itself in its own work and then spends potentially decades as the living intelligence network within the bone.
No construction project can proceed without clearing the old structure first. And in bone, the demolition crew is the osteoclast, one of the most remarkable and uniquely dangerous cells in the entire body. Osteoclasts are large, very large. They can be 50 to 100 micrometers across, making them among the biggest cells visible under the light microscope. They contain multiple nuclei, anywhere from 5 to 50, clearly visible within their pale, foamy cytoplasm. This multinucleated appearance is the single most important histological identification feature of the osteoclast, and it reflects their unusual origin. They are not derived from mesenchymal stem cells like osteoblasts, but from the fusion of monocyte and macrophage precursors of the hematopoietic lineage, driven by the signaling molecule rankle binding to its receptor rank on the osteoclast surface. Osteoclasts are always found adjacent to osteoblasts, working in a coordinated unit called the basic multicellular unit, or BMU. Their resorption process is directed by signals from osteocytes. When osteocytes detect mechanical stress, microfracture, or aging of the matrix, they reduce sclerostin secretion, thereby upregulating osteoblast activity and indirectly triggering osteoclast recruitment. Osteoclasts deploy targeted enzyme and acidic secretions, primarily hydrogen ions from a proton pump and cathepsin K, a cysteine protease, through their elaborately folded ruffled border, dissolving the hydroxyapatite mineral and digesting the collagen framework simultaneously. The released calcium and phosphate ions are transcytosed across the osteoclast and enter the bloodstream. Once the prescribed amount of bone has been resorbed, osteoclasts undergo programmed apoptosis, self-destruction. Their extraordinary destructive power cannot be left running unchecked. Bisphosphonate drugs for osteoporosis work precisely by inducing premature osteoclast apoptosis.
Everything we have learned about osteoblasts, osteocytes, and osteoclasts converges in the bone remodeling cycle, the continuous, lifelong process by which bone tissue is perpetually torn down and rebuilt, maintaining skeletal integrity, repairing microdamage, and regulating mineral homeostasis. The remodeling cycle operates in 4 sequential phases, endlessly repeating across thousands of microscopic sites throughout the skeleton simultaneously, visualized beautifully in this slide as a continuous infinity loop. Phase 1 is resorption. Osteoclasts, recruited to a specific site by osteocyte-derived signals, attach to the bone surface and excavate a microscopic cavity called a resorption lacuna or Hauschup's lacuna. The acidic, enzymatic microenvironment dissolves the mineralized matrix over approximately 2 to 4 weeks. Phase 2 is apoptosis. Once the required volume of bone has been removed, osteoclasts receive stop signals, primarily from osteoblast-derived effrin signals, and undergo programmed self-destruction, preventing over-resorption. Phase 3 is formation. Osteoblasts migrate into the resorption cavity and begin laying down fresh osteoid, filling the excavated space layer by layer. This phase takes significantly longer, approximately 3 to 4 months, because osteoblasts synthesize and deposit matrix far more slowly than osteoclasts resorb it. Phase 4 is mineralization. The newly deposited osteoid seam undergoes progressive mineralization as hydroxyapatite crystals nucleate and grow within the collagen framework, achieving full mechanical strength. This entire cycle, resorption, apoptosis, formation, mineralization, is continuously influenced by diet, physical activity, hormonal signals, and pathological conditions.
Now we arrive at the unifying clinical and physiological insight of this entire atlas, the principle that brings bone histology out of the laboratory and into the real world of exercise science, fracture prevention, and aging medicine. Bone is not a static archive of past construction. It is a dynamically adaptive tissue that constantly reads its mechanical environment and restructures itself in response. This principle is formalized as Wolff's law, the observation, first published by the German anatomist Julius Wolff in 1892, that bone remodels its internal architecture in direct proportion to the mechanical loads placed upon it. Here is how the response works at the cellular level. The stimulus: when you exercise, particularly when you carry weight, resistance training, running, jumping, you introduce mechanical stress into the skeletal architecture. Bone bends microscopically under these forces, and tiny cracks, microfractures, form within the matrix at heavily loaded sites. The sensor network, embedded osteocytes, connected through their canalicular pathways, detect this physical strain almost instantaneously. They respond by reducing sclerostin secretion, releasing the brake on bone formation, and by secreting prostaglandins and nitric oxide that recruit osteoblasts to the stressed zone. The response: directed by osteocyte signals, osteoclasts are recruited to clear the microdamaged matrix. This is immediately followed by osteoblasts laying down new, denser osteoid in the excavated space, producing a thicker, more mineralized lamellar structure capable of bearing greater loads. The insight for your patients and your own health: weight-bearing exercise is not merely muscular training. Every rep, every step, every loaded movement is a direct instruction to your osteocytes to build a stronger, denser skeleton. This is why weight-bearing exercise is the most powerful non-pharmacological intervention against osteoporosis.
We close with the most clinically significant bone disease in the world, osteoporosis. And using everything we have learned about bone cellular biology, we can now understand it with complete mechanistic precision. In the healthy skeleton, the remodeling cycle maintains a state of dynamic equilibrium. Osteoclast resorption and osteoblast formation are precisely balanced, and bone mass remains stable. Visualize a perfectly level scale: osteoclasts on one side, osteoblasts on the other. In osteoporosis, that balance is broken. The scale tips decisively toward resorption. Osteoclasts become hyperactive, demolishing bone matrix faster than osteoblasts can replace it. Simultaneously, osteoblast activity is reduced. The building rate falls behind the demolition rate. The net result is a progressive, cumulative loss of bone mass and structural integrity. At the microscopic level, this imbalance is immediately visible. The trabeculae of spongy bone, which should be thick, well-connected struts, become thinned, perforated, and eventually disconnected. Entire trabecular struts disappear. The cortical shell thins. The canalicular network of osteocytes becomes disrupted. The bone that remains is structurally compromised, capable of bearing far less load before fracturing. The clinical consequences are devastating. Osteoporotic fractures of the vertebral bodies cause progressive height loss and the corresponding characteristic kyphosis. Hip fractures in elderly patients carry a 20 to 30% one-year mortality rate. Wrist fractures from simple falls become common. Osteoporosis can occur at any age, but it is primarily associated with postmenopausal estrogen withdrawal, since estrogen normally suppresses Rank L and restrains osteoclast activity, and with secondary causes including glucocorticoid therapy, hyperthyroidism, and malnutrition. Treatment with bisphosphonates, denosumab, a Rank L antibody, or anabolic agents like teriparatide directly target the cellular imbalance we have described, restoring the equilibrium that living bone depends upon. This is what bone histology means in clinical practice.